Hello everyone. So we're going to continue on our discussion on lipids and biological membranes, but we're going to focus on membrane structure and assembly. If you take a look at the membrane itself, it's really impermeable to most polar charged solutes, and that's because you have these non-polar fatty acid chains, right? So it's mostly nonpolar, or it is nonpolar in nature, so it'll end up being impermeable to most polar or charged solutes. However, because of its nonpolar nature, it makes it permeable to nonpolar solutes. And if we consider the different proteins that are shown embedded within the bilayer, right? You have a number of different proteins that are shown embedded within the bilayer. If you also consider those proteins, this entire bilayer is typically anywhere between 5 and 8 nanometers thick, or up to 80 angstrom thick. Well, we're going to describe it in terms of the fluid mosaic model, which basically takes into account this lipid bilayer as the structural or the basic structural unit, and it gives a simplified and general image of how the membrane looks like. And this is done or came about based on a number of different studies. All right, so let's consider it based on the fluid mosaic model. What you have is different non-polar regions. You have different non-polar regions of the phospholipid, and those different non-polar regions, I'm just drawing over them in orange. You have these different non-polar regions. They basically form the bilayer face of the inner or the core of the bilayer. So they're facing each other away from water, away from the aqueous environment. Right, whereas the polar head groups are, facing the aqueous environment, which is the blue, and we've said that. Okay, so and then you have proteins that are embedded within the lipid bilayer, and those proteins as well have non-polar regions or residues that are basically in contact with the lipid bilayer, or hydrophobic domains that are basically in contact with the lipid bilayer, specifically with the fatty acyl chains of that membrane lipid, because we have this region of the lipid that is hydrophilic. So it's overall, if you think about it, is asymmetric. You have two different sides, right? You have the side that's facing the extracellular phase, the side that's facing the cytoplasm, and if you take a look at the proteins on either end, what it's attached to, you have two different sides. The protein domains exposed to either side are very different, and that really reflects the functional asymmetry of biological membranes. And you could think about the individual proteins and the lipids to form a mosaic, and that's where the fluid mosaic model comes in play. They form a mosaic that is fluid, and the reason we say that it's fluid is because most of these interactions with the protein interaction with the lipid bilayer are non-covalent. So the different lipids and the different proteins are really free to move laterally in plane of the membrane, right? They're really just going to be restricted by other proteins that are present on the membrane. And we had learned about the different proteins on a different video when we talked about membrane proteins. So very quickly, I guess we could talk about it. We saw integral membrane proteins, also known as intrinsic proteins. So that's an example of an integral membrane protein. But we also said that integral membrane proteins are tightly associated with the membrane through hydrophobic interactions. They could either be transmembrane protein, and that's an example of a transmembrane protein that spans the entire lipid bilayer, or they may be attached to only one side of the lipid bilayer. And those typically have nonpolar side chains that stabilize their interaction with the lipid bilayer. That's another example of an integral membrane protein right here. And we said they're typically alpha helical or beta sheets. Okay, and then we also have lipid anchored proteins, and we said that we have different types of lipid anchored proteins, whether they're prenylated, fatty acylated, or GPI linked proteins that are anchored to the membrane. And they could associate reversibly to the membrane, and that in response to biological signals. So they act as signaling molecules or are involved in signaling. And one example that we saw was palmitylation, right? And again, what we're talking about right now, if you did get lost, is lipid linked protein, right? That are anchored to the membrane by lipids. They can bind reversibly, and one example was palmitylation, and that anchor to the lipid bilayer was removed by an enzyme, if you recall, was known as palmityl thioesterase. That was on our previous video. Another example is phosphorylation of a membrane lipid. Phosphorylation of a protein that is anchored to the membrane by a lipid may trigger the lipid to either anchor to the membrane or the other way around, right? So it could be triggered by phosphorylation of a membrane lipid. And then we also saw the example of peripheral proteins that bind or associate loosely with the membrane. Right? That's just an example, right? It's electrostatic interaction. It could be hydrogen bonding with other integral membrane protein. And if you notice, all the oligosaccharides, the glycoproteins, are towards the extracellular surface of the membrane. So the fluid mosaic model was first proposed really in 1972. It was proposed by Singer and Nicholson, and then it evolved over time based on several different studies, and it explained one observation by Michael Edidin, all right, where two fused cultured cells intermingle. So what exactly are we talking about? Well, two different cells. One was a mouse cell and one was a human cell, and we're really explaining the observation of Michael Edidin, where you have two cells. Each of the different cells was fluorescently labeled with either a blue marker or a green marker in the mouse cell or a red marker in the human cell. And then they were treated with a virus, and where the two cells fused, and immediately after fusion, they were still segregated. You have the green fluorescent markers on one end from the mouse cell and the red fluorescent markers from the human on the other end. However, as time elapsed, those different fluorescent labeled markers fused basically or intermixed. So individual lipid molecules can really move laterally across the plane of the membrane by changing places with these neighboring lipid molecules, something we also refer to as Brownian motion. And we had seen lateral diffusion, right? So that's exactly what we're describing, but they're fluorescently labeled so we can actually see it, and it's a very rapid and can randomize the position of different molecules in really few seconds, right? Suppose the red one moves the other way around. So lateral diffusion can also be explained or shown experimentally by attaching a fluorescent probe to the lipid head groups, right? And then we use a fluorescence microscopy to follow the probe over time, right? So in one technique, what we do is we take this small region of the cell surface, and then we attach lipids, or we use fluorescence attached lipids, or we attach fluorescent markers to the lipids, right? And then we bleach that specific region by an intense laser radiation until that radiation region no longer fluoresces, so it completely bleaches it. It takes then, and then we wait for recovery, and it takes really only a few milliseconds for it to recover, where the unbleached molecules are diffusing towards the bleached area, and the bleached molecules are now diffusing out or away from it. All right, so we are looking at the fluorescent intensity over time and recovery, and the rate of the fluorescence to recover after this photo-bleaching is what we refer to as FRAP, fluorescence recovery after photo-bleaching, and it's a measure of the rate, really, of the lateral diffusion, right? It measures the rate of the lateral diffusion, right? But we're looking at lateral diffusion. And studies have shown that the rate of diffusion could be up to 1 micrometer per second. And again, we're just measuring the rate of lateral diffusion using fluorescence recovery after photobleaching, or FRAP. All right, so we're going to use erythrocyte membranes as an example to study membrane structure in assembly, right? Because they're relatively easy to isolate and overall simple. And we're going to specifically look at the different proteins that are embedded within the erythrocyte membrane. And we're going to start by looking at a protein known as spectrin, right? Spectrin is an alpha beta dimer, so it's showing you two, these are spectrin. I'm just gonna show one, but it's really an alpha beta dimer, right? So, you know, suppose this is the alpha, the beta is right next to it, right? It's an alpha beta dimer. Okay, so if this is alpha, this is beta, right? Anyways, it's an alpha beta dimer. It's a cytoskeletal protein, right? And if you notice, we had talked about glycophorin A, 131 amino acid residue, when we talked about transmembrane protein, erythrocyte glycophorin, right? So you have erythrocyte glycophorin, you have an anion channel. It's for really a chloride carbonate exchange. They're basically anchored or tethered to spectrin, right? So you have a number of different proteins that are embedded within the membrane that are in some sort restricted of their motion by either being directly associated with spectrin, right? By either being directly associated with spectrin. Why isn't it writing? Because it is, but that's really enough color. Right? You have some proteins that are tightly associated with spectrin. So what it's overall creating, think about it as fences that define regions within the membrane that restrict motion of whether it's lipids or different proteins within the membrane, right? So if we take a look simply at those two proteins, their motion is restricted within this particular region. And the same applies for the lipids as well. All right, and if we take a closer look at spectrin, it's an alpha beta dimer. Each of the dimers forms a triple-stranded alpha helical coil, right? It has a linker, and then what happens is that they further associate to form tetramer, right? And the alpha and the beta chain, their three-dimensional structure, or their tertiary structure, is fairly similar. All right, and so the cytoskeleton protein spectrum really is associated with various proteins that span the membrane and that are tethered to spectrin, right? One example is a protein known as incrin, right? You'll see a number of different incrins. If you take just a closer look at the different proteins known as incrins, they help spectrin associate or tether different proteins to the membrane, right? And incrin itself, well, it's a family of protein, but we're referring to incrin with an erythrocyte. It's a fatty acetylated acylate. It's a protein that is linked to the membrane by palmitoylation, right? So fatty acetylated protein. But it's also an integral membrane protein, right? So it's attached to the membrane by non-covalent linkage by a palmitoyl side chain. Okay. And if you take a look at spectrin, it's cross-linked at different regions, and we had just mentioned that a little bit ago. But you have this regions where it's actually linked to different complexes that contain the protein actin, right? So overall, what you have is a complex network of cross-linked spectrin molecules that are attached to the cytoplasmic face of the membrane, right? Which helps stabilize it. It helps it be resistant to deformation. And the different proteins that we see, protein 4.1, also known as band1, also known as Betty protein, I believe, they help regulate and stabilize the overall membrane structure. Okay, so we're not really looking into the specifications or the function or what would happen if there was a mutation in any of those proteins, but more so the overall structure of the membrane, right? So that's our focus. So proteins that you might want to remember the names of are like spectrin and perhaps actin and incrin, the ones that the book gives you more details about. And incrin is just a family of protein, and this incrin is really known as ANK1, right? So it's a protein composed of 33 different alpha helical residues, right? And just like the name implies, right, we said that it helps that the other proteins anchor through the membrane and associates with spectrin, and hence the name. Right, it helps anchor the different proteins between the membrane and the membrane skeleton, right? And if we consider the green part right here to be like the membrane cytoskeleton and A, B, and C to be different proteins, we could say that protein A interacts tightly with the cytoskeleton, right? Whereas protein C could more freely move and protein B is locked within a specific region of the cytoskeleton. That's just one way to rationalize, or envision how proteins might move around within the membrane. So just like the protein distribution is asymmetric, lipid distribution is asymmetric across the membrane. Cells have different mechanisms really that help control the type and the amount of membrane lipids. If you think about it, each domain, each tissue, each cell type, each organelle have different characteristic sets of membrane lipids, right? Okay, and those were initially determined by hydrolytic enzymes. The one we talked about was phospholipases that cleave at specific sites. And when membrane lipids are synthesized, they're synthesized on site. And this was determined through selective labeling techniques, where Kennedy and Rothman showed that membranes that are basically synthesized in the cytoplasmic phase of the membrane, or specifically phosphatidylethanolamine is synthesized in the cytoplasmic phase of the membrane. So what was done is that newly synthesized phosphatidylethanolamine phospholipids were labeled by a 1-minute radioactive pulse of phosphate. So this phosphate would end up being a radioactively labeled. And then if trinitrobenzene sulfonic acid, or TNBS, was added, it will interact with that phospholipids, right? Well, so you have newly synthesized phospholipids, you incubate that with radioactively labeled phosphate, and the ones that are radioactively labeled are actually shown in yellow. But we are about to make this yellow green. All right, so they're green. Okay, so it's just gonna use green. So anyways, for demonstration, so they're green. So incubated, they're radioactively labeled. They could be identified because they're radioactively labeled. And then if we immediately incubate it with the TNBS, it's going to combine with PE, but or the phospholipids, but none of the TNBS labeled phospholipids were radioactively labeled as well. So none of the ones that were radioactively labeled associated with TNBS. However, if some time allowed to elapse, in about 3 minutes, and then they were incubated with TNBS, then what they noticed that some of the ones that were radioactively labeled were also associated with TNBS, which came about the conclusion that PE phospholipids are synthesized in the cytoplasmic phase of the membrane, right? So, and then what's happening is a flip-flop diffusion, or a flip-flop rate, right? So, phospholipids that are synthesized on one side of the membrane can reach the other side very quickly. And this process is really facilitated by one or two membrane protein. The difference between the two proteins is that one is basically moving the phospholipid from one side of the lipid bilayer that has a high concentration to the side that has a low concentration, and the other one is so it's equilibrating the concentration of the phospholipid on either end, whereas the other type moves from the side that has low concentration to the side that has a high concentration. So one is known as flipases, right? So that one is tend to do the flip-flop of lipids from the side that has the high concentration of a particular lipid to the opposite side. So it ends up having an equal distribution of lipids. It's a form of what we're going to call later facilitated diffusion, right? And then phospholipid translocase is the opposite. It's moving it from its lower concentration to its higher concentration. So it's a non-equilibrium distribution, and it's what we're going to learn later as being what we call active transporter. All right, so the lipids, the protein, it's asymmetric. They can also aggregate, form what we call a microdomain, whether it's because of the presence of metal ions or other proteins, charged lipid head groups. Anyways, one example of microdomains is lipid rafts, right? Which are shown to consist of highly packed regions of sphingolipids and cholesterol. So what you need to keep in mind is within that um, lipid bilayer, there are different, it's asymmetric, right? It's not uniform in terms of the type of lipids that are present, the type of proteins that are present. So there might be regions that they're forming microdomains within the lipid bilayers, right? And we're talking about microdomains of lipids, the types of lipids that are basically forming those microdomains.